Good morning, everyone, and welcome to today's webinar, Understanding IVF, the Lab Process of Embryo Creation with Dr. Denny Sakkas with Boston IVF. Before we get started, I would like to go over a few quick items about today's event. First, all physicians in attendance will receive an AMA PRA Category 1 credit, and all other providers will receive a certificate of completion for 1 contact hour credit. The certificate will be emailed to the email you RSVP'd with by Harvard around July or August. Additionally, we encourage you to submit your questions to Dr. Sakkas using the question section of the control panel. You may send in your questions at any time during the presentation. We will collect these and address them during the question and answer session at the end of today's presentation. Now, without further ado, I would like to introduce today's speaker. Dr. Denny Sakkas is the Chief Scientific Officer at Boston IVF, where he actively cultivates new technologies that improve embryo development in our lab. Dr. Denny Sakkas received his undergraduate training at the University of Melbourne, Australia, and received his doctorate of philosophy at Monash University, Melbourne, Australia. Dr. Sakkas earned his PhD at one of the leading reproductive research units in the world. He has extensive experience in clinical IVF and reproductive research. He is internationally recognized, having organized numerous conferences and workshops on reproduction-related topics, and is frequently invited as a speaker at major conferences worldwide. He has published more than 250 peer-reviewed manuscripts and chapters in the field of fertilization, early embryo development, and male infertility. All right, we're going to get started. I'm going to pass it over to Dr. Sakkas.
Okay, thanks, Alyssa. Thank you, everyone, for attending today. And as we said, we're open to be questioned, or if you have any questions after this, or that are in particular related even to your patients, I'm happy to answer them. So today I'm going to try to have you understand a little bit about what goes in the lab and about sort of embryo development and pre-implantation embryo development. So hopefully I can answer some of the questions you might have about when you have patients coming through and what their embryos develop like. So the broad objectives, I'll talk about why not all embryos grow to a lovely baby or to a lovely blastocyst, at least in the in vitro process. What makes a good blastocyst? So the blastocyst is what we generally work with. It's about a day 5 embryo. And I'll also talk about freezing oocytes and embryos that we currently do. And if we have time, I can also answer some questions about how we might do it in the future. So hopefully you're aware of the IVF process. The clinical part is actually stimulating the patients to in some way exaggerate their IVF cycle so we can collect as many eggs as possible. Those mature eggs are then removed from the ovaries under ultrasound guidance and anesthetic. And then the lab part starts. So once we've got those eggs, we will fertilize them with the partner sperm or with the donor sperm using a technique called ICSI, where we inject the sperm straight into the egg, and I'll show you a video of that. Or we do something called conventional insemination, which was the original IVF process. The embryos, once they're fertilized, we actually grow them now up to about 7 days. So we give them 7 days to develop to what's called a blastocyst. And then if the embryos are developed properly, we either transfer that embryo back fresh, or we will freeze it for the future use and transfer them into the uterus. So the key steps in the IVF treatment cycle are we collect these eggs, which are surrounded by a cumulus oocyte complex. So these cells around them, obviously we collect the sperm, and then we grow the embryos. So day 1, so day 0 for us is the day of egg collection. Once we fertilize the embryos, we take them through and we actually check fertilization. And the way we check fertilization, you can see these 2 little sort of pools within the oocyte. That would be day 1. And then we follow the embryo as it grows to 2-cell, 4-cell, 8-cell. Day 4, it's compacted. And as I said before, sometimes we'll do what's called a fresh transfer on the 3rd day of development, or we will culture the embryo to the blastocyst stage, and we'll either freeze it for future use, or we can then transfer it also fresh for the patient if that's their choice. We've come a long way for the lab. In the old days, we used to use very large tubes, very big incubators. We mostly transferred embryos at the day 3 stage, and one of the problems was with that we weren't very confident in the way we grew embryos, so we actually would sometimes transfer 2 or 3 embryos. Hence, as you probably are aware, we would get a lot of multiple pregnancies, which was a huge black eye in the IVF world. Basically, the pediatricians used to hate us because we would, the MFMs used to hate us also because we'd actually give them a lot of multiple pregnancies. Luckily, now we're able to culture embryos to much later, to the blastocyst stage, and that's nearly all our patients here. But we now will normally transfer only 1 embryo back to patients in about 90% of our cases, and in general in the US, that's the number's quite high. So how does the IVF process work? So once we've stimulated the patient with cocktails of FSH and other hormones, we collect multiple oocytes. So the aim is for us at least, is to get as many oocytes as we can. When we collect the oocytes, when they come into the lab, they're not just nice and clean oocytes. They're oocytes that are surrounded by what's called cumulus cells. These cumulus cells are part of the sort of oocyte complex, and they actually feed the cells while they're in the developing ovary. And there's a lot of interaction that occurs between the oocyte and the surrounding cumulus cells, especially metabolic reactions and that. So once we've got those, we can do 2 things with them. So we can actually take those oocytes and we can inseminate them, so we can put sperm in straight away, or we can actually clean away all these cells and prepare them for another technique called ICSI. So this is and what we're actually looking for in the oocyte. We're actually looking to see that it's mature, and we can see that it's mature because it's got this polar body outside. So what we're looking for when we clean the oocytes is that they're in what's called the metaphase 2 stage, so they're ready to be fertilized. If we go back, this egg is immature, so we don't want these. But what we do get, about 80% of the eggs are actually mature and should be at the stage where they've actually extruded this polar body, which contains half the chromosome, so it's kicked out half the chromosome and it's ready for the sperm. So what we're looking for in about 90% of cases, 85% of cases, we see these types of eggs, but we do sometimes get some immature eggs. What do we do with these eggs? Well, we can do 2 things. We can either inseminate them, so just put sperm around, and the sperm will find their way through those cumulus cells that are surrounding the oocyte and actually fertilize the egg. Or in many cases, we will do what's called intracytoplasmic sperm injection. This is a technique where we take a single sperm and we actually inject it into the egg. And this was the first pregnancy was reported in 1992. This is an indication of how we do ICSI. You can see it's a very fine needle. This patient doesn't have many sperm, so probably doing conventional insemination, just putting in the sperm by itself and having them work it out would not work. So we catch a sperm, it goes into this very fine needle, so you can see the sperm coming in here. We then take that sperm and we will inject it into an oocyte. So you can see this oocyte, as I said before, it's got this polar body, so it's ready to be fertilized. So they're the eggs we look for when we're doing ICSI. We hold on to that egg, and then you'll see this actually magnify a bit higher now. Then we take that sperm and we will inject it into the egg. So this will just go into a bit higher magnification. You can see this is the polar body up at the top, the sperm actually going in, and you'll see the sperm will be injected directly into the oocyte. You'll see here. What we're actually aiming for is to actually put it into the cytoplasm of the egg. So you can see sometimes what we'll do is we'll aspirate some cytoplasm to make sure we've broken the cell wall here, the cytoplasm here, or the oolemma, and we'll inject that sperm into the egg. You can see the sperm into the egg in 20 minutes. It's amazing how fluid the membrane is of the egg. In 20 minutes, you won't even know where that sperm was injected. And what we do then, so this would be done, say, for example, today, and tomorrow morning, we actually would check those eggs to see if they're fertilized. And what we look for when we're checking for fertilization is something called pronuclei. So you can see these pools within the oocyte, which were definitely not there before. So one of these is the male pronuclei. and one of these is the female pronuclei. So that's how we tell, and this is very easy for us to see down a microscope and know that an egg has fertilized. And we expect about 70% of the eggs to fertilize to accept the sperm. Once it's fertilized, we obviously then will wait for the embryo to start growing. So within 20 hours, this egg that we see will become a 2-cell embryo. Then it'll start every 12 to 15 hours, cleave again to 4-cell, 8-cell, etc. Sometimes what we do see is abnormal fertilization, and you might get 3 pronuclei. So this actually means that 2 sperm have got in and 1 maternal nuclei. So these are abnormal, obviously. Interestingly, this they think is a forebearer of hytidiform moles, actually, that they're polyspermic eggs that have actually occurred in vivo too. So sometimes the block to fertilization doesn't work well, and you do get abnormal fertilization. So these we would not use for a patient, obviously. What we then do is we let the embryos grow, basically. So the embryos will grow. As I said, they'll go from 2-cell to 4-cell to 8-cell compacted. I'll show you a video a little bit later. But what we also follow sometimes is the way they grow. And what we're looking for in a good embryo is that the cells are growing in a uniform manner. So 2-cell, 4-cell, 8-cell. But that doesn't always occur in human embryos. We see a lot of variability in the way they grow, and sometimes an embryo will grow to the 4-cell, but it'll have some tiny fragments. So this is sometimes this is not that bad. Sometimes they'll actually have a lot of fragments. So you'll see a lot of fragments. So the embryos will not grow well. And this, unfortunately, we see in patients. Not all the embryos will grow perfectly. Sometimes embryos will grow to 3-cell instead of 2-cell. So we see a whole lot of variability in the way embryos grow. But the majority, we expect, if we're growing embryos to the blastocyst stage, we expect about at least half of them to get to the blastocyst stage. And that's one of the benefits of actually growing embryos for 5 days and not just 2 or 3 days. So this is just an example of various embryos. These are 3 or 4 days old. You can see, they're not all beautiful. Some of these are actually what they've undergone compaction, so the cells have already started joining. But you'll see a lot of variability in embryos between patients. This is a much nicer group of embryos, so you can see some of these are about 6 cells. 4 cells. These are sort of a bit more uniform, but sometimes we see a lot of heterogeneity, if you want, in the way the embryos grow. One of the things that we do struggle with is, though, we couldn't tell you here which of these embryos might actually lead to a baby. But we also wouldn't be able to tell you from these which embryo is likely to become a blastocyst. So that's why, in the majority of cases, at least in our lab, we will grow embryos to the blastocyst stage, because we believe the blastocyst is a better sorting tool for the good embryos. Because what happens here is I couldn't tell you, I've been doing embryology for many, many years. I really couldn't tell you 100% that which of these embryos would grow to a blastocyst stage. So why do embryos fail to grow? Well, there's some genetic reasons, metabolic reasons. They might not be metabolizing correctly. Maternal and paternal factors that can influence an embryo's development. In the majority of cases, it's a maternal factor. In some cases, a smaller percentage, it's due to the father or the paternal factor. But those all can actually affect an embryo. As I said, so having a brief lesson in embryology, say we were doing a day 3 transfer, we would actually probably transfer both of these embryos. But like I said before, these embryos look fairly similar, but one of them actually will only become a blastocyst. So, and really, this is why we culture embryos to blastocyst stage, because we're much better at we know a blastocyst is a much better embryo. So if you look at these 2 embryos, I really couldn't tell you which of these would be a blastocyst, but obviously one completely fails to develop and one becomes a nice blastocyst. So that's one of the reasons we actually grow embryos to the blastocyst. So the other way we do know is, if you follow these embryos, for example, we know that actually going to the blastocyst stage gives us much more information. So you can see these embryos are fertilized, all 3 of them, and they all grow a little bit differently. These are all from the same patient. Again, if I'm looking, this is about 40 hours, so this is the second day of development. If I'm looking to transfer these embryos on day 3, which will sort of come up in a minute, I can see the 3 embryos actually don't look that dissimilar. So it would be hard to pick which one would be the actual one I would transfer. So this is why sometimes we used to transfer 3 embryos, and you risk triplets. But you'll see here that even though these embryos sort of are growing in the same way, and you'll see in a minute they actually start compacting. They'll actually get all together, and the cells, we won't be able to count them, but you'll see that these cells here, some of these embryos, well, 2 of these embryos will be much better than another one. So you can see a few fragments here. These cells are breaking apart, but as the embryos develop, you can see this one here actually is a little bit ahead because it's coming together. It's at the compacted stage, and you'll see in a minute that this one actually degenerates completely, whereas it didn't look too bad on day 3. And you'll see a blastocyst forming in the other 2. Actually, this one actually looks much better. It's earlier blastocyst and is probably ahead of this one, which at one stage looked even much better. So these 2 we would freeze or we would transfer into baby. That one didn't jump out of the dish. We actually probably froze it. So that sort of gives you an idea of the differences in embryos. And we know we have the embryos for 5 days in the lab. There's a lot of things going on. There's morphological changes, there's genetic changes, there's metabolic changes, morphological changes. So there's a lot going on in the embryo that we really can't see in the early stages, and we believe that growing them to the blastocyst can sort out a lot of the problems that these have, that the embryo might have, and give us a better choice at which embryo is the best one. As I said, some of the things that do go on are metabolic changes. So we cater with these with the way we grow embryos, but we really can't look at metabolically, just as you would maybe measure the glucose in a patient. It's difficult for us to measure the glucose in a single embryo to know, although we're looking at these types of technologies to try to help us pick which embryos are the best ones. So right now, if we're selecting the best embryo, obviously one of the techniques we use is pre-implantation genetic testing. And this is a technology where we take a blastocyst and we take a few cells from the trophectoderm of that blastocyst as a surrogate as a marker genetically for that embryo. So you'll see here we remove a few cells from the embryo. Actually, the little red dot here is a laser. So we actually cut through these and we strip them away. So you'll see a few cells disappear into the pipette here. Again, remarkably, the embryo remains intact. So you can see that's the embryo here. We would freeze that embryo, and then the remaining cells we send for genetic testing. And this is a very robust test that we do. And we know that when we do this test that we can actually sort out the normal and the abnormal embryos at a fairly good frequency. We're very efficient doing it. And one of the better methodologies for picking embryos that are chromosomally normal in older patients, because you can see here, this is a graph of how many abnormal embryos there are. In younger patients, up to about 35, we only expect 20 to 30% to be abnormal. But you can see once a patient gets up over 38, the percentage of their abnormal embryos increases dramatically. So this technique of genetic testing is much more efficient at picking better embryos for older patients, but so they don't risk miscarriages in their pregnancy. So we can hopefully select the better embryos using this technology. One of the interesting things is that if you look at live birth rates in younger women, we expect now well over 50% of live births. This is an old graph, but we do know that this precipitously declines as patients get older. So picking a good blastocyst and picking a blastocyst that it's euploid is actually much better for the older patients, because once we have a euploid blastocyst, actually our pregnancy rates are just like if we find a blastocyst in an older patient that's euploid, the pregnancy rates are equivalent to finding it in younger patients. So we know also that young eggs, if we do donor eggs, we actually can transfer those back to patients at a very high efficiency. So it all goes back to the egg that gives you the quality of your embryo, of your blastocyst, and gives you a better chance at euploidy. So the younger the egg, the better chance we have of getting a euploid blastocyst. But what makes a good blastocyst? What do we look at in the lab that tells us that this might be a good blastocyst versus a bad one? This is an embryo developing. If you see the 2 cells here, you'll see the embryo at 2 cells. Again, it'll cleave to 4 cells. This is on the 2nd day of development. That's the time after fertilization down in the bottom right-hand corner. You can see this 4 cell will become an 8 cell. So this is a very well-behaved embryo. I wish all of them were like this. You can see that in a few seconds, this 8 cell embryo, these cells will compact and they'll become like a ball. So this is around the 4th day, and then we can't count the cells. So here we start relying on other features to tell us what's a good embryo versus a bad embryo. And you'll see as this embryo develops into a blastocyst, we will start looking at particular features. So you'll see the blastocyst, it'll have a cavity here, and you'll see 2 cells. You'll see some cells here that sort of are more ball-like structures, and you'll see the cells outside here. So what do we look for when we're trying to work out, is this a good blastocyst or is it a bad blastocyst? So we look for three things. We look to see how much, if the embryo is actually grown, if it's expanding. But the key things we look for, in the blastocyst, you now find 2 types of cells. What's called an inner cell mass cell. So this is a ball of cells that are down at one pole of the blastocyst. These actually go on to become the fetus or the baby. We also look at these trophectoderm cells. So these cells surrounding them, these very flat cells that have been pushed out by the fluid, are trophectoderm cells, and they go on to form the placenta. So what we've become very good at is actually assessing how good the trophectoderm cells are and how good the inner cell mass cells are. So the way we do that is we actually look to see how compacted the inner cell mass cells. So you can see here, these are inner cell mass cells. They should be like a ball of cells all sort of connected together. So we look at that, and we also look and see how the trophectoderm, the outer cells, are actually spread apart, if they're nice and flat or if they're uneven. For example, this inner cell mass cell here in this embryo, we really can't see a good ball of cells that represent the inner cell mass. You can see a few cells here, but they're all sort of spread apart, even though the trophectoderm doesn't look too bad. So this would, even though this is a blastocyst, we wouldn't consider this a very good blastocyst. And I'll show you how we actually score these in a second. So we look at a number of things. We look at how expanded this is, so how much volume it takes, and the greater the volume, the better it is. So if an embryo actually in our scoring system has a high value, it actually is a better embryo. But the key factors are 2 other things. So we obviously look at how well it's expanded. So you can see this embryo, which actually, interestingly, the blastocyst, because all the divisions are actually minimalistic, the blastocyst actually at this stage is the same size as an oocyte, as the big 1 cell that you saw before. But once we get this full blastocyst, what we look for is the inner cell mass cells and the trophectoderm cells. So we look and score how good these are. And I'll show you an example. So what we have is an ABC structure, if you want, for scoring embryos. So if the inner cell mass cells are actually nice and compacted, we will call that an A. If they're sort of compacted, but there's a few loose ones around, we'll call it a B. If they're very few, we'll call it a C. And if there's none, we will actually call it a D. So we actually grade these cells in relation to how tightly packed they are. We also do the same for the trophectoderm cells. So these cells that were on the outside of the embryo, if I go back here, all these cells on the outside here, we score those according to how compacted and how if they look like a kitchen tile structure, if you want, if they're nice and even and well spread out. So if there's many of these cells forming a very tight epithelial structure, it'll again be an A. If they're a bit looser, we'll call it a B. And if it's very loose, we'll call it a C. When embryos are very poor, we know that even if we freeze them, and we know the success of these types of embryos is very rare. They don't do very well. So if you're really interested in scoring embryos and you want to test yourselves, You can actually go to this site. We developed this site for ASRM, which is the American Society of Reproductive Medicine. You can actually go to this site and actually test yourself to look at different types of embryos and see if you can actually score them. So if you really want to understand this, this evaluation of blastocysts at least, you can try them out on this website. The embryo, though, is it's very tricky, and it's done a lot of things that we're not sure about, and there are ways of actually it corrects itself. So this is some work we've been doing in the lab where we can actually live image embryos. These are discarded embryos that are donated for research, and these we can actually look. And if you look at this blastocyst, you can see some cells here actually correcting themselves. So some of these were aneuploid. You can see this is the blastocyst. This is the cavity. These are all trophectoderm cells. And you can see, if I go back a second, you can see some of these. This is a cell undergoing mitosis. This is the daughter cells. You can see some of them actually correcting aneuploidy and actually going back to 2 or 3 cells and cleaving 3 times. So the embryo is also pretty smart at correcting itself, it appears, and has some tricks in its bag to make it actually more normal, if you want. What about freezing oocytes and embryos for the future? So this is the final part. So once we have good embryos, we're very efficient now at freezing those blastocysts, and I'll talk a bit about technologies that we use in the oocyte and for the blastocyst in freezing. So these are there are 2 ways we use to freeze, or we freeze embryos. One called slow freezing, and another technique which everyone does now. It's called rapid freezing. This is a technique called vitrification, and it allows eggs or embryos to be stored, and the success rates are the same or even better than if we had a fresh embryo, and there are reasons for that. In the old days, we used to have these instruments where we would actually put the embryos in these straws, and we'd freeze them slowly. So it would take hours to actually freeze an embryo because we'd sort of decline the temperature very slowly until we got down to liquid nitrogen. We would routinely keep embryos in these big liquid nitrogen vats. You can see here that there are little canisters here, or canes, and these are all numbered sequentially. We only ever put one embryo in one particular part. So this used to allow us to actually track embryos, but I'll show you actually how at Boston IVF now what we're using to actually store embryos. And obviously embryos are always stored in liquid nitrogen when they're frozen. So what's the difference between slow freezing and the technique we use now, which is called vitrification? So both have the same aim. We're trying to get embryos to in a solution at a temperature at around minus 200 degrees Celsius, which is liquid nitrogen. This isn't, I don't even know what it is in Fahrenheit, because I think in these terms, I think in Celsius, but this is very, very cold. It's like, this is where we can freeze embryos, we can store them for years, for 20, 30 years. Slow freezing takes you from room temperature or body temperature, but you can see it takes a long time, and very slowly you go down to this temperature. And we used to use an actually controlled instrument that actually allows us to get down to this temperature. Vitrification, what we use currently, is basically like these penguins dropping into the Arctic. You go from room temperature, and incredibly quickly, within a minute or 2, you actually go to, well, not even a minute, within seconds, you actually go down into this temperature. This system, vitrification, is much more efficient, and I'll talk a little bit about that in a second. So what are the difference between the slow and the vitrification? So what we do use in both situations, we use particular solutions to allow the embryo to be actually to be frozen. Many of you may not remember because you're probably too young, but in your car you would put a coolant in the radiator in the winter, especially in Boston or north of Boston, so it would not freeze. So not dissimilar, we actually exchange the water in this oocyte or the blastocyst, we suck that out, and we diffuse in what's called a cryoprotectant. So this could be something like glycerol, ethylene glycol, DMSO. There are a number of cryoprotectants. We have a special mix. So what we're doing is, in the oocyte or the blastocyst, we're removing the water and replacing it with these cryoprotectant or vitrification solutions. These solutions actually remove the water because it's the water that actually crystallizes. And if you can imagine these like sharp spikes coming in, and when it crystallizes, it will break up the cell or the oocyte. So the cryoprotectant doesn't do that. But before, in slow freezing, it wasn't as efficient as removing all the water from a cell. So you would still have some. So you would still actually have some crystallization within the cell. So even though this was a fairly efficient technique, it wasn't as efficient as vitrification. So vitrification, we actually really replace a lot of the watery solution with vitrifying solution, and we freeze it so quickly that we do ultra rapid cooling that there's no time allowed for crystallization to form. So you have actually a glass-like film within the cell, which is actually the vitrification. So that's why we believe the vitrification is much more efficient than actually slow freezing, which used to take a lot of time. They both end up in liquid nitrogen, but one is much more efficient than the other. So the sort of pros and cons of slow freezing, you use a slightly lower concentration of what are these cryoprotectants. It's longer. It takes much longer to perform. Technically, it's easier. And you need a freezing machine to do it. Whereas vitrification, you use slightly higher levels of these cryoprotectants. It's much shorter to perform and shorter exposure time. It's a bit more finicky if you want, but everyone here is really well trained and it works really efficiently. And you don't need a machine because you're just taking the device you're storing the embryo in and plunging it straight into the liquid nitrogen. So this is just an example. Vitrification is a process that produces what's called a glass-like solidification of living cells, not by crystallization, but by an extreme elevation of viscosity during the cooling. So you can see two fluids here. This is actually a vitrified solution, so you can see it's all clear, and this is actually a base medium that's frozen too. So the problem here is that you will have these crystals forming. So that's sort of the danger that you have for freezing using slow freezing versus cryoprotectant and vitrification. So this is just an example of what happens during vitrification and warming. So when we freeze anything, whether it's a blastocyst or an oocyte, it's taken through these solutions and plunged straight into liquid nitrogen. The warming of it, so once you take it out of the frozen, is the exact opposite. So you basically take the oocyte or the blastocyst, you put it in a culture media or a more dilute cryoprotectant. It replaces the cryoprotectant with the water slowly, so you're getting water going back in. You put it into the culture medium, and you have just a normal oocyte or blastocyst. So you can see here before vitrification, before freezing, you can see when you're putting in the equilibration solution or the vitrification solutions, the water's being sucked out and then replacing the water with these vitrified solutions. This is just after vitrification, but then it does swell up after vitrification. This then is plunged straight into liquid nitrogen, and then a month later, 2 years later, 5 years later, or 10 years later, you can warm the or thaw the embryo or oocyte, and you take it through the opposite process, and you basically have what you began with. So you can see here, sort of, there's really no difference between the one that was frozen versus the one before freezing. And we're seeing now, like, because of the advent of freezing oocytes and embryos, if you look at this curve here, so the blue line is embryos from frozen patient eggs or embryos. So this is the number of in thousands of treatments done. And you can see in early 2010, it was very few. It was much lower, the rate, because we weren't very confident of it. But you can see now, because of the confidence we've got in actually freezing eggs and embryos, you can see now the majority of treatment cycles we do from patients has grown immensely now, and this is up to 2022. It's even more now, that the majority of cycles we now do are with frozen eggs or frozen blastocysts. So we're really reliant on this technology now because of a number of reasons. And one of the reasons is that it's virtually more successful, or at least equivalent to if we were transferring embryos fresh. But one of the differences is if at the beginning I told you how we try to get as many eggs as possible, but doing that when we're trying to get as many eggs as possible, we're really changing the ovarian stimulation of a patient. So we're using very high levels of hormones. Now, if you imagine that when we're doing that, the uterus also in that patient is also getting affected in some way. So it's seeing higher levels of estradiol, fluctuating levels of progesterone. If we put an embryo back even 5 days after we've collected the eggs, that uterus is still probably in a hyper stimulated state, if you want. There are still a lot of hormones around. Even though it worked, we feel that actually divorcing the stimulation cycle from the transfer cycle allows that uterus to sort of calm down, be more like a natural cycle, and putting the embryo back during in a more relaxed and in a less stimulated, if you want, uterus gives us a better clinical outcome. And you can see now that frozen transfers now have really the number of frozen transfers we do and the percentage of live births we get is actually quite high, and even much higher than what we were expecting with fresh transfers. The other thing too is if we're doing genetic testing, or if we need to examine that blastocyst in some way, we can actually examine it. We can wait for the results to come back. It normally takes us about 1.5 to 2 weeks to get the genetic testing back for blastocysts. And then we know that which ones are normal, which ones are abnormal, and we can transfer them back in an ensuing cycle back to a calmer uterus, if you want. So I mentioned before, we also, when we're freezing eggs and embryos, obviously we're freezing a lot of these, we actually keep them in a system that's actually a bit like an Amazon pick station. I hate to say that, but it's all robotically controlled. We know exactly where an embryo is. The positions and that are all actually robotically assigned and actually documented in our computer systems, and actually are registered. And the freezing units we have these really big tanks that we keep embryos in. It's all monitored continuously, so the embryos, even though we've frozen them, they're as safe for as many years, and we know exactly where they are. So this obviously takes a really huge team to do it. In a busy clinic like ours, this is probably only about 2/3 of our embryologists. So we have a lot of embryologists that do this. They're all incredibly well trained at all the procedures I talked about. And so when you're sending your patients to us, the embryos are in the best hands as possible. I'll leave it at that. If you have any questions, you can email me at either of these two emails. The Boston IVF one, the top one's probably more common for you. So I'm happy to take any questions also, if there are any questions.
Thank you, Dr. Sakkas. We're now going to begin answering questions submitted during today's presentation. As a reminder, you can still submit questions through the questions pane in your attendee control panel. The first question is, can you talk a little bit more about embryo genetic testing, the different types there are available, and how it's performed?
Okay, so there are 3 types of testing that are performed. So the first one is genetic testing for aneuploidy. So this is purely looking for chromosomal abnormalities. The majority of our patients are sent to one particular clinic that is in our network called Juno. They're incredibly good at doing this. They've been doing it for many, many years. They use a technique called next generation sequencing. So we get a map of the whole chromosome content of the embryos. And basically what happens is you can see fluctuations in that map if an embryo is abnormal. It's a whole separate talk if you want, but that's screening for aneuploidy. So this is chromosomal abnormalities, Down syndrome, the most common ones are 16 abnormalities in chromosomes 16, 13, 21. So the similar ones that you would see in a recurrent pregnancy loss patient. So we're very good at screening these embryos. And as I said, unfortunately, you can't beat biology, and female age is actually very correlated to the numbers of embryos that you will have abnormal. So the the closer or around 40, you see a sort of an increase in the number of aneuploid embryos that we screen with these tests. So that's pre-implantation genetic testing for aneuploidy. The other ones we do is pre-implantation genetic testing for mutations. So this is looking at, so in the same technology virtually that we can look at any known single gene mutation in embryos. So we can actually test whether an embryo is affected, a carrier, or not affected. So say, for example, cystic fibrosis, which is one of the more common tests we do, we can tell if an embryo will carry or be affected by cystic fibrosis. So we can remove that from the pool of embryos and only transfer embryos that are not affected by cystic fibrosis. If the mutation is known, by the couple, we can actually test for it in nearly all cases. It's a bit more complicated to do this testing, and obviously the chances of having a normal embryo are driven by the balance of these genetic tests. But we can test virtually any embryo, whether it's for single gene mutation or some of the inherited disorders, even things like Parkinson's and adult onset diseases. We can test for. They're a bit more complicated, but I think it's one of the most powerful techniques we have in the laboratory. It's actually, unfortunately, it's not used as frequent, but single gene mutation testing is definitely something we can do, and it's very efficiently done with IVF. The third one is actually PGT for structural abnormalities in the chromosome. So if a patient has a translocation, a balanced translocation, we can again test that the embryo is not a carrier of this, or at least if it's balanced. So they're the three main tests we do. So I hope that answers your question.
Yes, thank you. The next question has to do with egg freezing versus embryo freezing and the differences in the labs for doing those, and maybe a little bit about the success rates.
So as I said, vitrification's really revolutionized the way we approach freezing. So both egg freezing and embryo freezing, what we put in, we virtually at 95% we expect to get out. So our success rates at actually warming embryos are well over 95%. So for an egg, we expect 95% of them to survive. Actually, for a blastocyst, we expect about 98% of them to survive. We're a very big lab, so we're doing a lot of these cases, a lot of egg freezing, a lot of blastocyst freezing. So we have very strict controls of who coming through the training for embryology, for example, who's allowed to do the egg and embryo freezing. And also when you've got one of your patients having their eggs or their embryos frozen, you can guarantee that embryologist has done a lot of cases and has a lot of experience. So, there are pros and cons to being a big lab, but one of the pros is that everyone in that lab is actually very well trained to do a lot of cases and has a lot of experience, whether it's freezing eggs, freezing blastocysts, whether they're doing ICSI, whether they're doing other techniques. That's one of sort of the bonuses, at least, of having a big lab, that everyone is super well trained. So to answer it briefly, when we're freezing eggs and embryos these days, we're virtually expecting the same results as we would with fresh, and like I said, even maybe a little bit better, especially for blastocysts.
Great, thank you so much. The next question is, do you prefer IVF than ICSI? I think they meant with ICSI. Do you prefer IVF with ICSI if there is no male factor, especially for unexplained infertility cases?
So it's actually a really good question. So conventional insemination, I presume you're you're asking. So the classic IVF technique compared to ICSI. So if a patient's a male factor, then obviously we're always going to do ICSI. Obviously in Massachusetts, because of the insurance mandate is, if the patient actually has above a certain amount of sperm, they're not covered for ICSI. So then we do probably around 45% of our cases are done with conventional insemination. So which means a lot of our non-male factor cases are actually getting conventional insemination. We actually published a paper. We published a series of papers, but we actually there's no difference in non-male factor patients whether you do conventional insemination or ICSI, and we actually tend to do more conventional insemination because in our own data we've actually found that actually it's slightly, we're talking about 1 or 2% better than actually if you did ICSI. One of the reasons, so let's say a patient has 10 eggs collected. Out of those 10 eggs, what we do if we're doing ICSI is we'll clean those eggs and we'll look for which ones are mature. Like I showed you, it's got the little polar body. So out of those 10 eggs, we would find 8 probably mature. So we would only treat 8 eggs for that patient. So we'll inject the 8 eggs. They'll get very good fertilization, very good blastocyst development. If we did conventional insemination, we don't see which ones are mature or not. We just put them in and we let the sperm work out overnight which ones sort of will fertilize. Every now and then, some of those immature eggs that we thought were not mature will actually mature overnight and get fertilized. So instead of maybe 10, you might actually have had 9 that were mature by the next day. And then, so every now and again, you'll get an extra one fertilized when you're doing conventional insemination. So you pick up 1 or 2 embryos every now and then for, every 3 or 4 patients. So we believe that actually IVF is actually probably better in some cases for these patients. The fear is you don't know if it's these are infertility patients. You don't know if ICSI would have helped them. But we also use a technique that if a patient has bad fertilization on the first day, we actually do a technique called rescue ICSI, where we will actually do ICSI the following day, and we have very good results with that. So even the fear of failed fertilization in these cases of conventional insemination, we sometimes it works in the patient's favor anyway. So we do both, to answer your question in very shortly.
Thank you. The next question is, what do you think about piezo-driven ICSI? Is it better than classic ICSI?
Shortly, no. Piezo-driven ICSI has become very popular in Japan. It's our ICSI fertilization rates are in the high 70%. So there's not much wiggle room to get better. In the papers I've seen with piezo-driven ICSI, in general, their normal ICSI rates are probably lower than what we would expect, and they're sort of getting up to what our rates are. So we haven't really looked at piezo-driven ICSI. We've looked at some other techniques to improve fertilization, but I definitely don't think, at least in our hands, piezo-driven ICSI is better than our classic ICSI.
Thank you, Dr. Sakkas. That was our last question. Thank you, Dr. Sakkas, and thank you everyone for attending today's webinar, Understanding IVF, the Lab Process of Embryo Creation. If you have any other questions, you can contact myself, Alyssa Cooper at ecooper@bostonivf.com. Once you leave today's webinar, you'll receive a survey on the presentation, and we would appreciate it if you could complete the survey and provide your feedback to help with future educational webinars. You will also receive a follow-up email within 24 to 48 hours with a link to view a recording of today's webinar. On behalf of Boston IVF and Dr. Denny Sakkas, thank you for joining us today and have a great rest of your day. Thank you, Dr. Sakkas.
Thank you. Bye.